All right, this will start the eukaryotic section of microbiology. First, we're going to start with the history. Approximately 2 billion years ago, we have first eukaryotic cells forming. This is through a process called endosymbiosis. Uh, endosymbiosis, well, first of all, you got to get the nucleus. And as you see with this particular cell right here, we believe it was an archaea cell that had an infolding of the plasma membrane that surrounded the DNA, creating a nucleus. Then over here, you have what a heterotrophic bacteria, one that uses is an aerobic bacteria, so it's using oxygen to create energy. And it's in the genus Rickettsia. We've tested the DNA, so we know this. It came to live it was engulfed by a larger archaea cell, as you see right here, and came to live inside of it. And they ended up, they became dependent upon each other. So they became symbiotic over here. But since it's inside of the cell, we call it endosymbiosis. And after a while, it lost some of its ability to live by itself. Now, most Rickettsia anyway are what we call obligate intracellular bacteria anyways, where they have to be inside of a cell to survive anyway. So it's not that far of a stretch to think about it. These eventually gave rise to mitochondria. So these here are going to give rise to all eukaryotes, and then they will branch off over here and become animal cells. This happened a second time with a cyanobacteria, right there. So these are photosynthetic ability, and this branch gave rise to algae and plants, things like that. Because they have the mitochondria, so we know the mitochondria had to happen first, and then chloroplast occurred second. So again, this is the origin of eukaryotic cells. In this picture here, the picture on the left, this is about a 900 million year old fossil found in rocks that are about 900 million years old. And you can see the fossilized chloroplasts inside of them. So, they've been around for about 1 billion years. The one here on the right, this one is found in China. You see the little spikes on it. They're actually all around it. This one resembles a modern day pediastrum algae. So again, these are evidence of ancient eukaryotes living. In here, we're going to talk a little bit about protozoas, some fungi, algae, helminths. We're not really going to talk about the arthropods other than the fact that they are vectors for diseases. In this particular drawing or picture here, this is not representing any one particular organism, because no one particular organism has all organelles. So like if it's got flagella, the odds are it doesn't have chloroplast, but it could. Then you have your nucleus, the nucleus, the nucleolus. Centrioles are primarily in animal cells, the glycocalyx, similar to prokaryotes and so forth. So this is just showing you all the possible organelles that are inside or outside of a cell, the eukaryotic cell. All right, the organization of this, again, we have the eukaryotic cell. We have external structures, which we'll go across. We have the boundary, which is mainly the plasma membrane, but there are cell walls. And then we have the internal organs, and we'll go over a few of these. Flagella, these are external and for movement. They have what we call the 9+2 arrangement. It's because they are in, there's 9 pairs around 2 individuals. And so that gives us our 9+2 arrangement. These are microtubules. They're quite a bit thicker than the prokaryotic flagella, and these are for movement. You have the whipping back and forth ones, and then you have these that they will swing forward, kind of grab the substrate and pull it forward or push it forward. Similar to each other, but they are slightly different methods of using the flagella. But flagella are for movement. Cilia are basically the same, except for the fact that they are a lot shorter, and there's many of these things on these. These are in a single group of protozoa and certain animal cells, primarily for motility and filtering. But like over here on this oral groove, the cilia can help get food into it for feeding purposes. They have the power stroke, which is where they're pushing back. And on the recovery stroke, they kind of lay to the side and swing right next to the surface. And then they'll come over and then go across, and then they'll swing to the side and then back across. And that's how cilia move. So it's a little different movement than flagella, but the purposes are very similar. The glycocalyx, this is our outer boundary, very similar to the glycocalyx on prokaryotes. It's mainly polysaccharides. It does have a network of fibers in there. It forms a slime layer or capsule very similar to the prokaryotes. It is for protection and adherence, again, just like in the prokaryotes. But another thing, except for whenever you're having your slime layers, it is for signal reception. So it's the communication from one cell to another cell. The fungi and algae have a thick cell wall along with it. And then protozoas, there's a few algae, and then animal cells lack the cell wall, which is going to come up next. Cell walls, that rigid structure, again, is for support and shape. Fungi have an inner polysaccharide, but their cell wall is mainly chitin. You have some with cellulose and some other glycans, but mainly chitin is what we think of for fungi. And algae and plants are primarily cellulose, but you have pectin and mannins. There is a group that have silicon dioxide. These are called diatoms. And then there's a group that have a calcium carbonate cell wall, and I believe they're called tests on certain species that have the calcium carbonate. The cell membrane, just like all cell membranes, no matter if it's a prokaryote or eukaryote, it is a bilayer of phospholipids and proteins. Now on eukaryotes, they have sterols, which helps make them stable. They are selectively permeable, so they're letting certain things in and out. Now eukaryotes also have the membrane-bound organelles. Prokaryotes do not have that, and this takes up more than half of the cell, of the internal, the inside of the cell. First check here, which eukaryotic cell structure is responsible for signaling between cells. If you remember what we were talking about, it was the glycocalyx. Just like in slime layers on prokaryotes, they communicate through that. For our internal structures, probably the most prominent one right there is the nucleus in eukaryotes. It is a double membrane, and it does have many pores in it so that things may pass through it. It's going to contain the chromosomes or the DNA, and it also has the nucleolus in it. And the nucleolus is what produces the ribosomal RNA and then forms the ribosomes, and then they get shipped out through the pores, either to the ER or out into the cytoplasm. Again, this stuff you should have had in a general biology class, so we're not going to go into a lot of detail on these. For the endoplasmic reticulum, this it originates from the outer membrane on the nuclear envelope. So it's connected to the nuclear membrane. All right, the rough ER has ribosomes. The smooth ER is without ribosomes. Ribosomes are making proteins, and it's the first step of the transportation process of it. So they make their ribosomes and they'll package them in what we call a vesicle to go to another organelle. Smooth ER is used to make and store lipids, and it's also important in detoxification. So it's going to remove kind of toxins, alcohols, things like that from the cell. That's what smooth ER does. They're very similar to each other, but they do have slightly different jobs. Here they're showing the outer nuclear membrane is synonymous with the rough ER you see, and then all the dots on it. It could be the smooth ER because they're showing you smooth ER over here. This is smooth ER. The ribosomes are on the outside of the cell. You have the inside. The inside is called the lumen. And inside of anything is usually called the lumen. And the outside is where the ribosomes are making the proteins. But we'll get to that when we talk about the ribosomes. The Golgi apparatus. This is our next one. This is it modifies, stores, and packages proteins. This is again with the transport system. It looks very similar to the endoplasmic reticulum, except that they have distinct layers. There's a layer, a layer, a layer. They're not continuous like on the ER. Now, important things about the Golgi is that it takes the vesicles from the ER, mainly this rough ER, but it could be smooth ER. And then it'll store these proteins that are made from the rough ER, and it'll modify them and mature those proteins and then package them. Then they put them into other vesicles to either transport them to organelles in the cell or to secrete outside of the cell. So for the transport, it goes from the nucleus, it makes the mRNA, which will go to the ribosomes to make the proteins, which go to the Golgi, they get stored and modified, they get packaged in vesicles, and I don't really necessarily like the secretion here because they could be secreted out like right here, like they're showing at this point, or these could be going somewhere over here to some other organelle inside the cell. So when I think of secretion, I think of it being transmitted outside, and it's not necessarily leaving the cell. Some of these vesicles that are inside of the cell, you have lysosomes, all right? These have enzymes, again, originating from the Golgi, and these are for the digestion of food or against invading microbes, because when phagocytes, your white blood cells, absorb bacteria, they put them into a vesicle. That vesicle will fuse with a lysosome. When it does that, we call it a phagosome. And then the lysosomes, those enzymes inside of that vesicle, will dissolve and break down the bacteria or the food if it's food. There's another set of vesicles that we call vacuoles, and these can store food or water for in microbes for times whenever there's not as much. These foods can be, again, for digestion, or it can be excreting waste. It just depends upon the environmental situations and what's going on. Mitochondria, we've mentioned them a little bit ago on the endosymbiotic deal. These are for energy production. All right, they have an inner and an outer membrane. The inner membrane is called the cristae, and you see in this artist's renditioning here, it's got lots of folds in it. If you look up in the micrograph up here, there you see the folds are very extensive. And the reason why is because the production of energy occurs on these, on the membrane. So the more surface area membrane you have, the more energy you can produce. Okay, and the cristae, so that inner membrane is where electron transport occurs, where we have our aerobic respiration producing lots of ATP. Now remember, we said that these are a Rickettsia genus because they have their own DNA, and it's circular like a prokaryote, not linear like a eukaryote. They have their own ribosomes, and it is a 70S ribosome. Eukaryotes have 80S ribosomes, so it's a different size. And they will divide independently. They do not need the cell to divide to tell them to divide. All right. So again, the prokaryotic DNA and ribosomes, what we're talking about here. Again, more evidence showing that they were another species that became that live inside of this the eukaryotic cells. Chloroplasts are the same way. Except in this, they change energy into chemical energy through photosynthesis. So there's the green ones. We find them in algae and plants. Again, an inner and an outer membrane. The inner membrane is folded up into these thylakoids and then it's stacked into the granums. Granum is plural. This is where the photosynthesis occurs, is in these areas. All right. They have their own DNA, just like a mitochondria does. They reproduce independently, just like mitochondria do. They have their own ribosomes, again, 70S. Again, all this is evidence pointing to these being cyanobacteria in origin, and they just lost some of their abilities to live on their own, and they live inside the eukaryotic cells. These, again, are primary producers for nutrients and for other organisms. Ribosomes, these are producing proteins. So they are made up of ribosomal RNA and globular proteins. They're scattered in the cytoplasm and on the rough ER. They're larger than prokaryotic ones. Remember I said that these are the 80S size. Your mRNA will go through these, and multiple ribosomes, we call it polyribosomals, can be using that same mRNA to produce more than one protein at a time. The ribosomes are on the outside of the ER, and the proteins get transmitted inside the ER. It's calling it the cisterna, but in the previous pages, they call it the lumen, because lumen is inside of anything. Inside of a tube. And that's how it gets packaged and then sent on to the Golgi apparatus. So again, these are for protein synthesis. Cytoskeleton, two main parts are the microfilaments and the microtubules. These are both of them are involved in the movement of cytoplasm around in the cell. So you have we call it cyclosis. And then they're used for the amoeboid movement, which is still the movement of the cytoplasm, making the projections on the cell membrane. So they transport, they help move things through the cell, and then they give it structural support. That's all we need to know for this one from this introductory class. Oops, sorry. This table in your book, so table 5.2, shows you the genetics, reproduction abilities, respiration, all this stuff of organisms. Notice over here on the viruses, they do have, let me get my pen. They do have their own nucleic acids, but everything else is negative because they don't respire. They don't photosynthesize. They don't move around. There's no biochemicals going on, no reproduction. That's why viruses are considered to not be alive. Your prokaryotes, when you have the plus minus, I mean, some can, some don't, depends on what they are. And look at the eukaryotes. The eukaryotes do a lot more than the prokaryotes because they have more capabilities. But I wanted to point out that the viruses don't hardly do anything. Some have a membrane, we call that the envelope on them, if you remember that. All right, our next one, next check. The eukaryotic organelle that is responsible for transporting vesicles inside the cell. This one is the Golgi for transporting in. Remember, rough ER makes proteins, it goes there, gets packaged, and then either moves it to other organelles or outside of the cell. We're getting into the taxonomy now real quick. Taxonomy, I don't think there's any one set that really anybody agrees on. I mean, that we have some big things like everybody agrees what animals are, fungi are, plants are. And we know that moneras are the prokaryotes. But this group right down here, these are all the protists. And actually right here, this one was in the protist also. But doing ribosomal RNA analysis, we said, wait a second, green algae and the cryptomonads are actually more closely related to plants. So they're getting changed and put into this group. The protist kingdom was kind of a catch-all kingdom. If it didn't fit into something else, they just kind of threw it in there by their appearance and their morphology. But now that we can do genetic testing on them, we see that no, these don't necessarily agree with everything. Because I believe, no, that was not. So they're breaking them down into more groups. So the protist kingdom, we're still going to use that term, but it's getting broke down into different super groups or whatever you want to call them. So taxonomy is going through a makeover right now. And I do not envy the people doing that because it's a tedious job to determine all of that. But I just want you to know that it is under revision, but we're going to use kind of the traditional kingdoms for this. And since all of these are under the eukaryote domain or eukaryote domain, we're going to that, and then we'll break them down to their kingdoms and so forth. All right, so our microbes, fungi would be the first ones we talk about in the kingdom fungi. Two main types, your macroscopic and your microscopic. Your macroscopic are your mushrooms, puffballs, and gill fungi. Your microscopic are your molds and yeasts. By far, the majority of both groups are unicellular or colonial. And then some of them have the cellular specializations, and that's the macro ones up here. But by far, the most of them are unicellular. For the morphologies of these, you can have the round yeast or the filamentous hyphae. And then there's some that are dimorphic that form both, either the hyphae and the spores. And we'll go over a lot of this. All fungi are heterotrophic, and until the 1960s, I believe they were in the plant kingdom, and plants are all photosynthetic. So that was a breakthrough there. Again, the majority are harmless. Fungi are saprobes living off dead plants and animals. Some can be parasites. They're usually what we call opportunistic parasites or opportunistic pathogens, meaning that they can infect something if it's weakened or in the right conditions. For like us, it's usually, well, here, athlete's foot, it's got to have the right conditions for it to grow there. But other fungal infections usually have to have some kind of a weakened immune system, like it's an AIDS patient or a cancer patient or somebody whose immune system is compromised. Any fungal infections is called a mycosis. And fungi are everywhere. They grow in snow and on glaciers, so they can survive the cold. They survive in the deserts. So you're not getting away from fungi. They're all just all over the place. All right, so the organization of these yeast, these are soft things. Soft looking when you see them in their groups. And this is usually it's an asexual process. If they are yeast, they're asexual process through budding. We'll get into some other stuff in a moment. Your filamentous ones. All right, so these have hyphae, and when you look at hyphae, that's going to be cottony or velvety or hairy or look like it's got a velvety texture to it. Some hyphae are separated with a septae, as you see right through here, and others are non-hyphenated, septated. I'm not sure how to say that. We have two main forms of vegetative ones. These are the ones that are just kind of growing happy-go-lucky, digesting food, absorbing their nutrients, just going about their business. And then you have what we call the reproductive ones, and these are the ones that are going to produce spores for reproduction. All right, so reproduction, most of them are going to go through spores on the reproductive hyphae. We do have asexual reproduction on these things, and those can be through either budding or mitosis or on the Conidia sporangiospores. All right, so now sporangiospore goes in the sporangium, and sporangium is a closed a sac that has those sporangiospores in them. Conidia, Conidia, these do not have that sac containing those spores, whether it's an arthrospore, chlamydospore, phyllospore, whatever. These do not have a sac containing those, unlike the sporangiospores that are stored in that sac first. And that's the difference between a sporangiospore and a conidia. Sexual reproduction on these spores is, again, it's the fusion of two different strains. All right, we have three main ones that we're going to talk about: the zygospores, ascospores, and basidiospores. And these are sexual spores, so they're going to form these spores through the sexual production. We're going to break them down here. So in this case, we're looking at the zygospores. You have two different organisms. They have the plus and minus. We can call them male, female, it doesn't matter. They will merge together and form the zygospore. And this eventually will form the spores to be released from the sporangium. Now this form with the zygospores, these are diploid. So the spores in the zygospore process are diploid, okay? So they're 2N, they have a full chromosome count. And now they have, it's half the chromosomes of this one, half the chromosomes of this one. So there is some genetic variability in this to help them better survive. And, you know, for evolution go on. Over here on the ascospores, again, we have the male and the female spores, excuse me, hyphae. They merge and you get the new hyphae coming up out of it. Excuse me. And up at the tips, which is what we're drawing, looking right here. Inside here, you have the spores in here. Now, unlike the zygospores, these are haploid. But it is from two different species, not species, but two different specimens. So it is a mixing of the genomes, again, for sexual reproduction. And then you release those spores, those ascospores, but they are haploid, so chromosome count of N. Basidiospores are much the same way. You have your plus and your minus, male, female, however you want to say it. These will grow, they merge together, and they will grow. And when they're ready for the mating, you have the mushroom what we call a mushroom merge from the ground, because the hyphae are under the ground, and it's a mushroom that emerges is the reproductive structure. And this happens to be a gill mushroom, and these gills is where the spores, in this case, the basidiospores, are going to be released. Again, these are haploid, just like in the ascospores, but it is from two different individuals that merge together to make that. So this is a reproductive, this is sexual reproduction to produce these spores. And again, these are haploid to go on and spread the genes that way. So it's a mixture of the genes. So we classify these. There's several ways to do it. We're classifying, our book does it on their sexual reproduction. So zygospores, and these are mainly the sporangiospores, some conidia. Then you have your ascospores, which are all conidia. Basidiospores are all conidia. There's a couple of more phylums in here, the Chytridomycota ones. These have flagellated spores, or flagellated spores, yeah. And then you have some that are only asexual. We call those the imperfect fungi. Again, this is just a brief information about all fungi. All right, so how do we identify them? Well, first of all, we have to isolate and grow them. Auger for growing on them, so you can grow them on cornmeal, some blood auger. Saborod's is a very common one for growing fungi. Then once you grow it, you look at the macroscopic and microscopic observations of it, just like we would with prokaryotes. So we look at their asexual spore forming structures. We look at the type of hyphae they have. We look at what the colony looks like, the colors we have in it, physiological characteristics, so what they can consume, what they can't consume. And in the last 20, 30 years, maybe, genetic makeup's been a lot easier to do. So now we can actually do genetic identification of these in most labs. Adverse impacts. Again, most fungi are not, they're not parasites, but they can be opportunistic. So again, people with weakened immune system like AIDS, cancer, other immune problems, they can become, they're susceptible to getting fungal infections. They can cause allergies, causing your body to overreact. Some produce toxins. Aspergillus is a black mold that has a potentially lethal toxin that it can secrete. It can cause problems in animals and humans for that matter. Fungi, since they are saprobes, they're eating dead things, and fruit and seeds are I guess they're not really dead unless they're viable, but they will decompose those. So it's estimated that roughly 40% of your fruit crops are lost to mold every year. So it's a big economical impact in farming industry, agriculture. Benefits of these. Again, they do decompose dead plants and animals and recycle those nutrients back into the soil for other organisms to grow. We get a lot of antibiotics from this. Our first antibiotic, penicillin, is from a fungi. Produce alcohol. Now these alcohols can be for industrial or it could be for consumption like wine and beer. They'll produce a lot of organic acids and they can produce vitamins. We use fungi not just in the industrial settings like this, but we use them to make cheeses. We use them to make beer, beer, but breads. When you cure meats, they'll add flavors to meats, so cured that can affect the flavor of that. And they use them for genetic studies because they can be sexual and asexual reproduction on them. Here's a list in your textbook of different groups, and some of them, they give you some non-pathogenic ones and some pathogenic ones in here. All right, fungi are generally classified according to their, since they can do both the sexual and asexual, we don't classify them by that, and they're everywhere, so we don't really classify them by habitat, so it is by the hyphae that we classify them. All right, now we're on to kingdom Protista, which is where most of these are going to fall under. All right, we have algae and protozoa. Algae are, these are all photosynthetic and they use chlorophyll A, which is different than the chlorophyll that we find in prokaryotes. And then protozoa are all unicellular, and so that means they lack tissue because there's only one cell, and they have similarities in all this other stuff, okay? Again, most of these are unicellular, some are colonial on the algae. Again, algae are photosynthetic. They can move around. Like this here is an example of a Chlamydomonas, and you can see that it has two flagella, but it does have the chloroplasts in there. This is a single-celled organism. Some of them can be multicellular or colonial. This example down here is these are diatoms, and this is that silicon dioxide shells that they make. So it's basically a glass shell. And all of these are going to have a cell wall depending on what it's made of. And some have flagella, some do not. Okay, but they do contain the chloroplast and do the chlorophyll. They have other colors besides just green chloroplast. And that's how you get different colors of your algae. These are, by far the majority of these are free living. We don't normally get any illnesses from these, but they can, mainly on the dinoflagellates, because if when you have runoff, And you can have what we call a bloom, a red tide, or and they because of the pigments in them, they call it gives them a red color. And these give off toxins that will kill fish and other organisms. But it can, your filter feeders like your clams and mussels and whatnot like that, they will filter through this and it builds up in them. And if people eat that, those organisms that have been contaminated with this, they can have neurological problems or even die from it. And it takes several months for these toxins to be filtered out of the marine organisms. One very important thing here is that algae, what we call plankton, produce a very large amount of the atmospheric oxygen, and they're very important for that. So we need to treat them with respect on it because we need them. But they're also a big source of food for they're the base layer of food for aquatic habitats too. Here they're giving you a list of these various types of them based on their pigments in the cell walls. Notice that they are used a lot in cosmetics. We use them as food additives, and there are some medical products from these. Protozoa, several tens of thousands of these things. Again, key things on protozoas, they do not have a cell wall. By far, the majority of them are unicellular. I can't even think of a colony one off the top of my head. Most are harmless, okay? Their habitats are limited by moisture. They do cause some parasites, and they can be spread by insects. This is where we throw insects in there. These are all heterotrophic, so there's no chloroplasts in these things. And they feed much the same way as our white blood cells consume bacteria, as by engulfing microbes or other organic matter. A lot of them go through these trophocyte and cyst stages. So again, many of these have flagella to move around or move around through pseudopods. And they go through the trophocyte cyst stage. So the trophocyte, this is the one where they're feeding. They're actually moving around doing stuff. When the environment gets bad or conditions get bad, they will form a cyst, which will help protect them, kind of like an endospore on a bacterium, but it's not that hardy. All right. And when the conditions get better again, then the cyst will it'll start growing, and they'll go back to the trophocyte and start moving around again. They reproduce asexually, but they can produce sexually through conjugation, very similar to prokaryotes, except that prokaryotes conjugation is genome moving one direction. In the protozoa, it's moving in both directions. Classification is difficult because there are so many of these things. Here we're kind of grouping them on their motility or their reproduction. Mastigophoria, again, flagella, sexual reproduction. Sarcodina, primarily the amoeba type movement, asexual. Ciliophora, these are the ones with cilia. These are the ones that go through the trophocyte and cyst stage, most of them. And then apicomplex, and these are all parasitic here. Okay, so just a few things on them. Here's Mastigophoria. This species right over on the right, this is Giardia. It has a feeding group here. There's flagella on it. This looks Euglena over here. No, because Euglena has chloroplast, so it's not a Euglena. But it looks very similar to a Euglena. I'm not sure what that particular one is. Just a general, they're calling it a paranemia. Over here, you have the Sarcodina. So this is the amoebas. You see the cytoplasm and all the organelles in them, and they'll produce what we call the pseudopods, where they kind of they have extensions that reach out and pull the organism along, and then they engulf food. Over here, we have a Blepharisma. Blepharisma is the Ciliophora, so they have lots of cilia for moving around. There's an oral groove in here. The cilia help give food into them right there, microbes that they eat. This particular one on the apicomplexa, this is looks like a Plasmodium. I'm not 100% sure. These are parasitics. Here's a list of ones that are parasitic and that cause infections. We're going to cover a few more. The first ones here, some prominent ones are Trypanosoma. There's two main species of these, the Brucei and the Cruzi. They both look like this shape, this kind of folded leaf shape. To me, it looks like a folded leaf, crinkled up leaf. This one here is the Chagas disease that we're talking about here out of South America. Now, African sleeping sickness is treatable, but if it's not treatable, it will cause death within a few years. Chagas disease, this is from the kissing bug. They'll come up by your mouth and bite you, but you don't get infected from the bite. It's their feces that has the protists in them, and when you scratch at it, because it'll cause you to itch, you kind of break the skin, and that's how the protozoa gets into the skin, and then it gets spread into your pets, or it can be that can be spread from wild animals. Again, it's that spread in the feces, and it does cause scratching, so that's how you get a break in the skin, and it gets spread that direction and that way. The biggest cause of dysentery or diarrhea worldwide, and actually kills lots of people because they're in less industrialized areas where they don't have medical treatment for it, is Entamoeba histolytica. You get it from consuming the host, either the cysts in either contaminated food or water. So you go in, they hatch or go to the trophocyte phase. They're going to lay more eggs, which cysts, excuse me, not eggs, which is excreted in the feces. The feces, because their sanitation is not as good in unindustrial countries, or people just don't wash their hands, so it gets transmitted that way. But a lot of times the feces gets into the water. They use the water to irrigate stuff, but so then it gets contaminated on the food, and then you consume it in that aspect, or you're just, again, consuming the contaminated water. That's how we get it. It is treatable. You just have to have the medical facilities to do it. All right, what's the following descriptions of both algae and protozoa? Well, only algae are photosynthetic. So on that egg base right here, we can go down to D. Only the protozoa are heterotrophic, so that eliminates those two. We know that they're not always unicellular. All right, so that means it's got to be C, that they both do contain mitochondria, and that's because both of them are eukaryotes. We're almost done. We're on to the helminths now. These are multicellular animals. Okay, so again, they're animals. They have very extensive organs for reproduction. They do have some digestive organs, some movement organs, and they have some means of protection. The reproduction was a big one. We're just going to be talking about the parasites on these. So they were going to host in the host tissues. They have mouth parts for attachment and to digest. Again, that well-developed sex organs to produce the eggs and sperm, because that's pretty much what these things do. They go through a larval period. It could be in or outside of the host, just depending upon the species. The flatworms are two different types. So we have flatworms and the roundworms. The flatworms, there's no definitive body cavity on these, and it's simple excretory or nervous. All right. We have these get broke down into tapeworms and flukes. Okay. Then we have our roundworms, which have a complete digestive tract and a protective surface, but their excretory and nervous systems, again, are poorly developed. Some examples of these, which is how we're going to classify them. We have our cestodes, which are the tapeworms, and all of these segments are pretty much just sacs for eggs, because that's their biggest thing. They just produce lots and lots of eggs. On your trematodes, which is your fluke worms over here, notice that a lot of these organisms here are hermaphroditic. They have both female and male reproductive systems. And look at it right here. It takes up almost half of the body, because they're very developed, because that's what these things do. They just produce lots and lots and lots of eggs to spread around because that's really all they're worried about doing. About 50 of them parasitize humans. They are worldwide, but they're mainly in your tropical and subtropical areas because that's where you're going to have a higher incidence of it because of the warmer climate. You get these from ingestion of contaminated food or water. If you have open sores, you can get them in. You can get them from the soil or being in water, and they can be carried by insects and spread that way. Afflicts billions of people a year because you can get multiple infections of it. Here's an example of one that affects, there's no socioeconomic status that protects you from pinworms or region, because these are everywhere. Children tend to get it more so than adults because they're not as clean. What it is they'll consume it. It'll go through their oral route, call it the oral fecal route, because it gets in the mouth. Eventually the female will move to the anus where they lay eggs, and it causes it to itch. So they're scratching, which is what they're showing you right over here. I can draw this. And so then it gets on the hands, and then the hands will infect clothes. You can pass it to other people and affect your bedding. So usually, but not always, if a child in a house gets it, it's likely that everybody else in the house has it also. It takes about a month for the female to start laying the eggs from it, and somewhere in that range. But again, it's very treatable, so it's not a big deal. And it just happens just because children aren't as clean as adults are. They don't wash their hands as much, and they put more things in their mouth. That is it for our eukaryotes. Have a good day.